Welcome to the fourth lecture for Bio 1104. In this lecture we're going to be looking at cell structure and function. And the references there for you. Please, some people have been looking on StudyDesk and trying to access those e-resources. We think we've got the links fixed now, but if you are having trouble, that might mean that someone else is looking at it at that time, so come back a little bit later, okay? So in this lecture we're going to be looking at some of the major differences between prokaryotes and eukaryotes. I spoke about this last week, the basic differences, and you had, if you remember, those three domains, and we'll be exploring that a little bit more, and then some of the shapes and characteristics of certain bacteria. Some of the internal and external structures, that's important to know, because how does the flagella actually work? How can some bacteria move that don't have obvious flagella? So how does that work? So we'll be going through a little bit of that and looking at bacterial cell division, because it's not as intuitive as you would think. But if we look, just simplistically, if we think about one cell, say E. coli, because it's the one that can generate every 20 minutes. If we think of one E. coli cell that undergoes binary fission every 20 minutes, so at 20 minutes there's 2 cells, at 40 minutes there's 4 cells, yes? What about after 12 hours? 24 hours. We'll be looking at growth characteristics too. Not in this lecture, but in other lectures we'll be looking at how the growth actually, the trajectory of the growth. So I want you to be able to distinguish or explain the characteristics of prokaryotes. How do they actually differ from eukaryotes? And it's quite, there's just a few very simple differences. And look at the structure and function, as I said. The movement mechanisms of bacteria, so we're going to be looking at how that flagella actually work, and look at some of the structures inside the cell wall, so of the cell wall, and then internal to the cell wall as well. And so, if we think about cell types, there's 2 distinct types of cells. We've got the prokaryotes, which are the simple cells, and then the eukaryotes, which are more complex cells. Now, the prokaryotes are unicellular, so there's only one cell. But the eukaryotes, sorry, I'm not explaining that very well. So prokaryotes are single cells, and how we define prokaryote is we look at their nucleic acids. Okay, are they in a nucleus or in that general vicinity? Okay, so it's kind of spread throughout the cell. And so karyon means nucleus, or more correctly, nut. Okay, so it doesn't have that nucleus where the chromosomes are kept. But the eukaryotes do. They've got a definite nucleus, okay, and that's where the chromosomes are kept there. So all prokaryotes are single cell and all are bacteria. And eukaryotes are plants, animals, fungi, protists, so they're much more complex. And you'll remember this diagram from last week. So we have the bacteria, the archaea, and the eukarya. So these, once again, are the more complex. These bacteria, the prokaryotes, are the simpler, and the archaea are the ones that have characteristics of both kingdoms. Okay, sorry, both domains. And if you can remember that the archaea are the ones that can live in extreme conditions as well. Now, we had a look at some images, stained images of cells in the last lecture, but just to go into a little bit more detail what those cell shapes actually look like and how we describe them. So if it's spherical, you have, it's known as a coccus. One coccus or many cocci. Okay, so singular, plural. And cocci just means berries. So if you think about staphylococci, then it's a group like berries. Now bacillus just means it's rod-shaped. A bacillus is the singular, bacilli is the plural. Spirillum is spiral, so you kind of get used to the terms. But, and there's always a but, these can change shapes depending on the environment, their age, and competing bacteria. Some can even change form. There can be a virulent form and a non-virulent form. So if it changes a form, it might it might go to a cocci or something, and the cocci form is the one that produces the disease. Okay, so they can change shape. And as I said before with E. coli, sometimes if you look at it quickly, you might think that it's a coccus, where in fact it's a very short bacillus. And so then that's known as a coccobacillus. Okay, if it's kind of in between. So we've got the coccus, the bacillus, and a coccobacillus, so that's the intermediate form. Then we've got one that looks a bit like a comma, which is known as a vibrio form. Then we've got a spirillum, so it's just a wavy cell. And then we've got another one that looks very similar, but if you think about it in 3D, it's like a slinky, you know? So it moves, it spirals around. Okay, that's the spirochete. And they can be motile as well. So the shapes of bacterial cells are important to know. And their arrangement, once you see the shape under a microscope, you can also look at the arrangement. So are they single cells, or are they diplococci or diplobacilli? Do they form pairs under the microscope or in your smears? Or do they form chains? Okay. It's very, why they would be in pairs or forming chains, it just means that how they're dividing. So when they divide, they might not completely divide. They might stick together. So if you are dividing in one plane, you can get Diplococcus or Streptococcus. Okay? Or you could have tetrads. So you've got a picture of a tetrad here. That just means that it's dividing in two planes, okay, to form that tetrad. Then you have the Sarciniae. These are the ones that will reproduce in three planes, but they don't disengage from the cells, from the other cells. So they form little groups, clusters. Then we've got another type of cluster form, which is the Staphylococci. And it looks more like a cluster of grapes, like a grapes. Then we have the bacilli. These can be in chains, as I said, or they could form rosettes. Now that's really weird, because what they do is they are basically tethered to a substance. And so they're tethered, and they look like a rosette. So they can be in lots of different types of arrangements. And even some of the archaea, so the really extremophiles, can form star shapes or even squares. So there's a number of different forms. But the size of bacteria, why we need that compound microscope, they're typically quite small. So between 0.2 and 1 micron in diameter. There are a few exceptions which we'll be looking at a bit later. And the reason that they're so small is that means that they've got a large surface area to volume ratio, and that allows rapid uptake of nutrients. Okay, so it's the surface to volume ratio. Not only can those small cells actually take in nutrients, they can get rid of waste really easily, because you don't want waste to be building up in the cell itself. And the smallest bacterial cells are below about 0.2 micron. But if we go from the very small to the very large, well, large, we've got a cell that is quite different. It's actually Epilopiscium, I think it is, fish lysone. It is found in the gut of sturgeonfish. Now, it will be active for, I think it's during the light cycle, and when it's active, it actually changes the pH in the sturgeonfish gut. So it can dissolve or activate the enzymes to break down the algae that they feed on. And then in the dark cycle, those bacteria are actually not active, and so the pH varies accordingly. These are quite large, and they elongate, so they look, I don't know, like a long tube. And they, instead of undergoing binary fission to reproduce, they actually produce spores. So they're very weird bacteria. But there's another type of bacteria that's also very large. Now, where the other one was elongate, this one is a coccoid form. But with that coccoid form, they have little granules inside. And when it was known, Phytomargarita namibiensis, it's known as that because when the light hits it, those granules in the cell are actually sulfur granules. And when the light hits it, they glow like a chain of, and they typically grow in chains. So it's like a chain of pearls, hence the name. They also will produce, so they're found in sediment, in rivers, so bodies of water, they're found in the sediment. So not only do they store the sulfur, but they can actually use some of the nitrates as well as some of their food source. Okay, so they are really, really interesting large bacteria. Now, I've called them extra large because if you look at the volume, they're probably larger than the elongate ones that we had a look at before. But it's very rare for sulfur to be stored in vesicles. Now, cell structure. I'm saying that it's a very simple cell, and you can see that it is, but it's got a lot of components to a prokaryote. So if we start with the chromosome, maybe we'll start with the list, sorry, from the outside in. So we've got the glycocalyx. This is the structure around the cell wall. So typically it could be a capsule or a slime layer, depending on how closely linked it is to the cell wall. Then we've got the flagella, or the flagellum. So you can see that it's got like an anchor here, and that allows, that spinning motion actually allows the flagellum to move and the cell to become motile. Okay. Then we've got the, it doesn't have there, the pilus. Now the pilus, remember I said that a pilus chromosomes can be exchanged, so genetic material can be exchanged from one cell to another. There are specific types of pilus or pili. So these pili are just these extensions of the cell. So there's special types of pili that are known as conjugation pili that are formed between two cells and genetic material is exchanged. But if they're smaller, if they're less elongate, so these types of ones, these just a very short hair-like structure, they are the ones that can allow the cells to stick, to adhere to surfaces. They're made of the same material, but some are very long, some are very short. Okay. Then we've got the cell wall that we'll look at in a little bit more detail. And then underneath, just deep to that cell wall, we have the cell membrane that you're all familiar with. That phospholipid bilayer. Yes? Remember? Okay. Then inside the plasma membrane, we've got the cytoplasm. So that's just that fluid material where you have the chromosomes floating around and different types of inclusions, including sulfur granules if it's one of those extra large bacteria. So lots of vesicles that you find in there. You also have ribosomes. Where are we? With ribosomes for protein synthesis, and you can also have plasmids. So that extra DNA, circular DNA that can be found in there. And these plasmids you'll be learning about in a little bit more detail when Louisa comes to chat. And so the inclusions are the other vesicles, etc. And sorry, I'll just draw your attention to these intracellular membranes as well. So in a few more slides, I'll just show you why those intracellular membranes are important. They actually come from the cell membrane, but they're incorporated differently internal to the plasma membrane, or to the cell membrane. So it's a very interesting way of using it. But if we start with the glycocalyx, so the outside bit, so it has, and when you do your pracs, you'll be looking at different plates, and you can see these are colonies on an agar plate, and you can see that they're kind of really glossy, kind of slimy, maybe slimy, glossy, we'll go with glossy. Glossy and mucoid is the term. And this is indicative of those cells all having a covering around the cell themselves, which is polysaccharide, so a glycocalyx or a capsule. And those capsules or slime layers, is it a capsule, is it a slime layer? If it adheres quite strongly to the cell, it's a capsule. If it can be washed away quite easily, it's a slime layer. Okay, that's how you define it. And why are they important? Well, they can allow, it's one of those virulence factors. If it's sticky, it's going to stick to epithelial cells, and then it can colonize. It can also, that glycocalyx can also prevent desiccation, so drying out. So it's a good preventative as well. And the capsules are not found in all bacteria, and the composition of a capsule can change between species as well. So typically we don't look at the composition of capsules. We look, if we're using classical techniques, we're looking at the presence or absence of them. And as I said, because it's a virulence factor, not only can it help to adhere to surfaces, but when you've got that covering, it stops phagocytes engulfing it as well. So it can evade your immune system. And so Streptococcus pneumoniae is a capsulated bacteria. Sorry. And here you can see a cell. So we've got the background, the negative stain, and we've got the cell staining, but the capsule surrounding it doesn't stain. Okay, so it's quite easy to see where there's capsules. Okay. Now, a slime layer, this is associated with strep mutans, and so dental caries, for example, so there's the plaque buildup. And typically, you will have a certain type of cell or species actually adhering to a surface, and they produce different a slime layer, for example, to protect themselves. Okay. Then when they protect themselves, what can happen is other bacterial types come and join into that colony. Okay, so it's a mixture of different bacteria, and they can get quite large, but they still have that protective layer around it. And when it gets too large, what can happen is a few of them just basically slough off and go and form the start of another biofilm. Now, biofilms are very difficult to get rid of because, you can probably see this, you might get rid of the top layer, but there's still more there. Okay, to be able to produce another slime layer. And this can be quite problematic because with the biofilm formation, it's not only on your teeth. It might be in a urinary catheter, a bladder catheter. And when if they're motile, that biofilm can move, okay, and cause disease in organs. Okay, so biofilm formation is quite nasty. So not just teeth, it might be in a hip that's been replaced as well, or a bladder catheter. So there's a number of different ways that biofilms can be medically important. Now, if we think about movement, because if you're a cell, a bacterial cell, you've grown, you've reproduced a lot, you've taken in nutrients from the environment, you've gotten rid of your waste, but the waste is starting to build up. So the environment is becoming quite hostile. If you don't have a mechanism to move out of that environment, that cell is going to die. Yes? Unless it's got endospores, and it will produce endospores. But if that cell has a flagella or a means of motility, then it can move from a hostile environment to a new environment. And that's where flagella come into their own, because they can move from one point to another point, typically with nutrients, but sometimes there are other reasons as well. So the structure, and this is quite nice. If you look at the electron micrograph here, you can see the structures of the flagella. Okay, so in the cell here, and then if we look at it in the image here, this is just a schematic of what that actually is. So you can see there's a number of different anchoring points in the cell membrane and in the cell wall as well. And then this hook actually will rotate, okay, because you have to have some sort of rotation for the flagellum to work, yes? And when you do a serology of a bacteria, for example, you can test for what type of antigens are on those flagella. So that's where you can get the H is the H antigen is the flagella antigen, whereas the O is for the body, the main cell. And so it's got a hook and a basal body. That's how a flagella works. So typically we find them on the outside of the cell. And they can be in a number of different arrangements. If we have, if you look in A, this is a monotrichous flagellum. And in B, this is lophotrichous. Amphitrichous is one at each end. And then the peritrichous, so flagella all around. But they always work in unison, because if they didn't, it would just go nowhere, okay, if it's got more than one. So those flagella always work in unison to move from point A to point B. And they do that by simple flexing. So they can have a rotation or a corkscrew motion, and typically most or many bacilli have flagella, but typically most cocci don't have flagella. And I'm sorry, you're going to have to get used to that wavering, that qualification of mostly, because there are some exceptions. And if we're talking about movement, we're talking about taxis. So that movement can be toward a chemical or away from a chemical. And phototaxis can be movement toward a light source or away from a light source. So chemotaxis and phototaxis are some of the terms that you need to be aware of. And the phototaxis is, you might see that when you're doing your motility, your hanging drop preparation in the lab as well. Now the bacterial cells that don't have extracellular flagella, they can still move as well. So there are internal flagella, so the axial filaments. So if you look at this scanning electron micrograph, and it's been colorized, so don't worry about the color, but it's just been colorized so you can see this yellow circular bit or spiral inside the cell. Can everyone see that? And that is the axial filaments being stained or coloured. Now then, if we look at this cross-section here, we've got the axial filaments around one side, and you can see that they move around the cell. That is a wonderful arrangement because these are also endoflagella. That means that the flagella is inside. And because they circle around the cell, just underneath the cell, the plasma membrane or the cell membrane, when they move, they cause the cell to spiral as it's moving. So it's a flagella inside the cell. Now we get to the pilus or the pili. These are, as I said, hair-like appendages, and they're composed of something known as pilin and allow for adhesion to surfaces. And as you can see with the conjugation pilus, it's quite elongate and you can move. So remember the plasmid, that circular bit of DNA? It, in whole or in part, can be moved through this conjugation pilus to another cell. The scary part is it doesn't have to be a cell of the same species. So if you had an antibiotic resistance gene on that plasmid, it may be transported or exchanged, or transported is probably a better word, to another type of cell. Okay? And you can see these other hair-like appendages. So these are the fimbria. And they are much shorter than pili, but allow that adhesion to surfaces, as I said before. Now, the bacterial cell wall, we've looked at that, and this is just another way of looking at it. And so you can see a very, bad typo. In the cell here, we're looking at that in that square, you've got the lipid bilayer, and then you've got the peptidoglycan, okay? And it is quite complex, and you can see what it's composed of there. So don't worry too much about that. It would be good to know what those particular substances are, but you don't need to know the exact structure. And this is just showing the difference between gram-negative and gram-positive, and they have, the different bacteria have different cross-linking molecules as well. But what is more important, I think, is to see it visually. So if you have a gram-positive bacteria, you've got that large layer or very wide layer of peptidoglycan, and you've got, with all of these, you've got lots of proteins associated with that. And what you've also got is the, so that's the gram-positive. So what colour would it stain with gram stain? Gram-positive is purple. Positive purple. Then we've got gram-negative bacteria. So it's not nearly as large, that peptidoglycan layer, but what you can see is on that outside, the outer membrane, you've got a lot of proteins in the phospholipid bilayer, and you've got these things called lipopolysaccharide. Can you see that? So little appendages to the outside, proteins. Now, lipopolysaccharide, you may have heard of LPS. It's shortened to LPS, and that is one of the toxins. So if you have an infection with a gram-negative bacteria, that LPS on the outside of the cell can cause an inflammatory response and an increase in temperature. Okay, so it's a pyrogen, an increase in temperature. Then we've got the acid-fast bacteria. I thought we'd just put that in because you can compare the staining techniques, and you can see that it's got a very wide lipid layer before you get to the peptidoglycan or the cell membrane. So that accounts for the difference in staining. Yes? Okay. So if we look once again at gram staining, and I'm going over this again because it is important, you've got the differences. The stain distinguishes between cell wall composition. So with the gram-positive, so gram-positive is purple, you've got the bacteria retaining that crystal violet and iodine complex. Remember, when they're joined together, they form a much larger molecule, and therefore can't be washed away very easily. And they have a thicker peptidoglycan, so it's about 90% of that wall. And you've got teichoic acid present in the cell wall as well. Now, the gram-negative organism don't retain the crystal violet iodine complex. It gets washed away. They've got that thinner peptidoglycan. That's why they don't retain it. And then you've got the outer, on the outer surface, you've got lipopolysaccharide or LPS. And that actually, not only will it increase temperature with an infection, but it prevents entry of some antibiotics as well. So this is quite a nice table because it goes through the gram-positive, gram-negative and acid-fast bacteria, just giving you the characteristic that's in all of them, okay? And you've got some type organisms as well. So Staphylococcus aureus, gram-positive, Escherichia coli, gram-negative, and Mycobacterium tuberculosis as an acid-fast. Okay? Now, if you look at sensitivity to dyes and antibiotics, so gram-positive are the most sensitive, gram-negative are moderately sensitive, and the acid-fast are the least sensitive. So you can understand why some treatment for this might be problematic because it's they may not be terribly sensitive to antibiotics or dyes. And with antibiotics, how do they work? So remember gram-positive bacteria? They've got a very thick peptidoglycan layer, yes? Antibiotics, such as penicillin, actually block that peptidoglycan synthesis. And what happens if you block that? Then the rigidity and the integrity of the cell is destroyed. And so you get the destruction of cells. So that's why they're more effective against the the gram-positive bacteria. Lysozyme is one of those enzymes that you can find in a lot of different secretions. So saliva, tears, mucus, they, also the innate immune system, they all produce lysozyme or contain lysozyme. And that in itself can help with discouraging bacteria and bacterial growth because it breaks down or digests the peptidoglycan as well. Okay, so you're destroying the integrity of the cell. And everyone should remember the plasma membrane or the cell membrane. You've got the phospholipid bilayer, and not only that, you've got a lot of proteins in or transmembrane or just stuck on either side, those peripheral proteins. They can be channels, they can be antigens for different things. They distinguish self from non-self as well. So you can look at the different antigens to determine what type of bacteria it can be. So the O antigens, for example. Now, if we go inside the cell membrane, we've got the cytoplasm, nuclear region, the ribosomes where proteins are produced, and the intracellular membranes as well as inclusions. So the cytoplasm, what can you say? It's semi-fluid and it's inside the cell membrane. Okay, that's good. No questions about that? But that is really important because everything else floats in that cytoplasm. Other things are transported through the cytoplasm. So it is very, I'm sorry, I shouldn't have been flippant about that, but it is a very important substance in the cell itself. Now, then we have the nuclear region, and you can see that it's a region. This is where the chromosomes tend to aggregate, but they're not enclosed in a particular or a discrete nucleus. So it's a nuclear region. And typically you have a single bacterial chromosome packaged in that area. But you could have plasmids as well. And we saw that in that circular DNA, and that is where you can find genes for antibiotic resistance and a lot of other things as well. Now with the ribosomes, this is really interesting because ribosomes are all about making protein, yeah? Eukaryotic cells have ribosomes as well as prokaryotic cells. Yep. So prokaryotes, though, have a different type of ribosome. It is known as a 70S ribosome. Now, S is for the Svedberg or sedimentation rate, so it's just basically the size. And it's composed, that 70S ribosome is composed of a large subunit and a small subunit. And no, I haven't made a mistake. They are those different sizes. So the 50S and the 30S together form a 70S. I know. Yes, I know. It doesn't make sense, but that's how it works. Okay, so it's just when they're joined together, it's all about the sedimentation rate. So they sediment at different rates. It's not my maths. And so in the ribosomes, you get the translation of mRNA into those polypeptides. So you have the protein synthesis. But if you wanted to get rid of particular bacteria, what you could do, and leave the eukaryotic cells, your own cells, alone, what you could do is just target that 70S ribosome, couldn't you? If you took that out of play, then there would be no protein synthesis in that cell and it would die. And you would leave your cells alone. They shouldn't be affected. And I'm saying shouldn't because you always have to remember that there's the mitochondria in your own cells as well. So it's always a bit of a trade-off. Now, I spoke about intracellular membranes before. So those internal membranes, these come off, originally they're butted off from the cell membrane, but they're internalized. And with some of those intracellular membranes, they can be used. There's a number of changes in them, and so they are associated with chromatophores, and that's where you find them in photosynthetic bacteria and cyanobacteria, for example, and they use the sunlight and photosynthesis to grow, to produce their nutrients. Okay, so their energy. So and it's a really beautiful image because you can see those photosynthetic membranes and the carbohydrate molecules as well. Okay, so that's photosynthesis. Now, you've got other soil. So typically these are the chromatophores. Chromatophores are in bacteria that are in mostly in water, in fluid. But you also have some of the bacteria in soil that have intracellular membranes as well. And they don't photosynthesize as such. They actually oxidize. There's enzymes there that oxidize nitrogen compounds to produce nutrients for the cell. So those intracellular membranes are quite interesting. Now, I spoke before about inclusions, so those vesicles that are found inside or in the cytoplasm itself, and they can function as reserves of nutrients, so storage. So it, with that large, the extra large bacteria, they have a very large vacuole in the middle of it that stores nitrates. So storage vesicles are quite good. So they can be, those reserves can be either granules or vesicles, and the granules aren't bound by a membrane, whereas the vesicles are. Now, some of these vesicles, some of the bacteria can store other types of things. And it was, I think in the 1900s, a scientist was looking at bacteria under a microscope and recognized that they were forming a line at specific areas. They were always forming the same sort of line. And that was, it was discovered, but it was actually explained a few years later by these bacteria actually having magnetite in their granules or their vesicles that actually were aligning to the magnetic poles. Okay, so it doesn't have to be sulfur or anything like that. It can be things like magnetite. I think it's really interesting. Like, what would be the advantage of being able to align to a pole or to be magnetized? Anywho, now endospores, these are the ones that can be formed by some bacteria, not all bacteria. And you can see quite nicely where the endospores are in the cells. It could be identified using a gram stain or a simple stain and looking for a void, or you could use a special endospore stain where the endospore actually takes up the stain. But it has to be really, has to be heated for about 5 minutes for the endospore to take up the stain. Now these are typically formed, you generally find them in older cultures, because in older cultures that means that they have used up a lot of the nutrients in that particular area and they're undergoing stress. Okay, so what they do then is they put all of their genetic material into an area, one particular area, and then wall it off. And then if the environment continues to be harsh, then the vegetative cell around it will die and you're left just with the endospore. Now, that endospore wall is really tough, okay? But if it gets into an environment where it's favourable for growth, then it can germinate and start growing. Okay, so have you ever trodden on something or you had a piercing wound and you had to go and get a tetanus injection? Yeah? Well, why would you have a tetanus injection? Because Clostridium tetani is one of these endospore forming bacteria. And if you had a piercing wound and it could be contaminated by soil, that's where you could find endospores. And they can live for over 100 years in soil. So chances are, hopefully, you don't have an endospore inserted into your body. But if you did, it's about 37 degrees. You've got lots of protein, lots of fluid, lots of nutrients, a beautiful environment to start to grow so that endospore can germinate and you might have a tetanus starting to, a tetanus infection. And that's why you have that tetanus injection. So what does the tetanus do? So that Clostridium tetani, it produces a toxin. And that toxin affects your nerves and the neurotransmitters particularly. And if you have seen, there's a horrendous picture of someone from the 1600s or something like that who has tetanus, and all of their muscles in their body spasmed so much that they broke their spine. So That's the sort of thing. That's why if you have a piercing wound and someone says get a tetanus injection, say yes. Because the tetanus injection just means that if there's any toxin being produced, that injection will actually mop that toxin up. Okay, that's what it is. And yeah, so mainly Bacillus and Clostridium, especially soil bacteria. Now with the bacterial cell division, this is known as binary fission. Okay, just very, very simply, you get this, the chromosomes, so the genetic material replicating and moving to either side of the end of the cell. And then you get the cell coming down, forming a transverse septum. And when it is complete, then the cells, those daughter cells, separate if you're getting single cells. Okay? But you have to be aware that everything in the cytoplasm will replicate as well. So you get movement of everything that you need in the cell. And that septum actually comes down and kind of pinches them apart. So we've looked at the bacterial shapes and some of the ones that are less common as well, so the square and the star shape and the rosettes. And we've looked at some of the names and functions of the main parts of a prokaryotic cell. You understand the difference between prokaryotic and eukaryotic. And we've looked at some of the movement or motility of the cells. We've also looked at the composition of cell walls, so that adds to the gram-positive, gram-negative conversation, and looked at bacterial cell division. So, if you've still got a few minutes, which of the following is not a bacterial cell shape? C? Everyone happy with C? Okay, what about the next one? The antibiotic penicillin is most effective in killing. D? Any raise on D? Exactly right. So thank you very much, and I will see you in the prac tomorrow.